Okay, good morning and hello everybody. Welcome to the third day of our symposium. We're going to start the morning session with an invited speech from our dear colleague Ian Max Andolina, which I've had the pleasure of being introduced to I think about now 6 months ago or something. So I'll give a brief introduction for Ian, and then we're gonna basically be all ears to listen to his speech. Ian currently is an associate investigator and director of cognitive and behavioral analysis platform at the Institute of Neuroscience in Chinese Academy of Sciences, CAS. He did his Bachelor of Science in Neuroscience and University of Sussex, and a PhD at UCL with Adam Salido. And he also did his postdoc with Adam, working on the mechanisms of cortical and cortical-thalamic feedback from higher to lower visual areas. His current research interests are focused on visual processing in human and non-human primates, as well as artificial visual systems. As part of a broader initiative at the newly founded International Center for Primate Neurobiology in Shanghai, he is also developing novel cognitive and behavioral tests for transgenic monkey models, which sounds very interesting. So today's talk, as you can see, is titled The Neural Priors That Guide Visual Illusions, and I think we're all ready for you, Ian, to start your speech. Thank you.
Great, thank you. So just to confirm that you can still hear me?
Yes, we can hear you.
Okay, excellent. Great. So first off, it's a great pleasure actually to be invited to give a talk at this symposium. It's actually very impressive, and I think you guys have done a really good job. So I would like to thank the organisers, and I think, as Ali said initially, that it's been organised by students. So particularly thanks to the students. I think you guys have done a really, really impressive job. So this is why I'm very, very happy to speak here today. Okay, I'm going to start my talk about 2,500 years ago, and it starts with Plato's Allegory of the Cave. So for those who don't know, this is a very famous allegory that was found in Plato's Republic. Effectively what happens is, you can imagine that there are a group of humans who are slaves or prisoners, and they're trapped in a cave, and they basically are chained to a floor, and all they can see is a wall on the opposite side, and they see these forms, weird forms and shapes, and they live their whole life in this cave, bolted down to the floor. And the shapes that they see actually are shadows, which are being created from a light source of fire, and so all that they can perceive are basically sort of shadows and sort of copies of forms as they really are. And Plato uses this to imagine what would happen if one of these prisoners was suddenly let free, and they walked out of the cave into daylight. First off, they would be blinded. The question is whether they would recognize the things that they saw, these shadowy illusions in the cave, whether they would understand that they were the same things that they saw in the cave or not. And Plato then goes on to say that the person who is in this space, now liberated, sees the world for what it really is effectively the philosopher. And so, Plato's idea is that you should teach philosophy to everyone. They should become enlightened and see things as they really are. And so, for the purpose of this talk, the idea is that actually Plato assumes that most perception is incorrect. The perceptual sensations are all illusions. So everything that you see in this case it is an illusion, and you need to use cognition, you need to use reason to try to find out what the true forms of things are. This created a debate and an argument, and the interesting thing is that the debate that was created is the same debate that's going on in perceptual neuroscience now. So there was a very famous skeptic, Diogenes, and he argued against Plato. Plato had this idea of these ideal forms. So there's a cup that you see, but actually there's a perfect or an ideal form of a cup which you can only access through logic and your rationality. And so Diogenes says to Plato, I can see a table and a cup before me, but I don't see the tableness or the cupness. There's nothing in perception which, gives me this sort of, meta object. And Plato responds by saying, well, it's natural enough. You've got eyes, so you can contemplate the things that come into your visual system, but actually you also need intellect, and that's where the tableness and the cupness are seen. And what this is really a sort of a discussion of is, whether conscious perception, awareness for perceptual things out there in the real world is, sort of low level or high level. And, whether the perception that we have is trustable. And this is an argument which basically has rolled on since its time through Kant and Hume, all the way through to in the 2000s. There was a big debate between Steve Koslin and Zenon Pilchnin on the content of imagination. When you imagine things, what's going on in the brain, and right through to today, where there's a big discussion at the moment on the concepts of cognitive penetrability, how much cognition can influence perception. These are all sorts of parts of the same story, and ancient philosophers thought about vision in sort of one of two ways. One was a theory which is sort of shown on the left here. It's called the theory of extramission, and it sounds a little bit ridiculous to us today, but actually it was a very good theory at the time. And the idea is such that you actually have light in your eye, a sort of a fire in the eye, and the fire is projected out into the real world. And almost like the sense of touch, you palpate the things in the real world using the light that comes from your eyes. And this idea carried on for well over 1,000 years. And actually even today, very interestingly, people still feel that the gaze of the other has a sort of a strong impact. And there was a paper a few months ago in PNAS that actually showed that if you show people looking at an object and you look at the eye movement of the observer looking at a picture of someone looking at something, they make eye movements consistent with this sort of ray that is emitted from the eye. But the idea that we sort of are used to now is the idea of intromission. So basically something comes into the sense perception. So again, this is a sort of a medieval illustration, and you can see here that you have the eye connects forwards, in this case, the frontal cortex. In fact, it's not even the cortex, it's the ventricles. This is the ventricular theory. So the actual cortex and thalamus and all the other stuff was a sort of a padding for the important thing, which was the ventricles in the brain. And so you had sort of perception here, and you went on to do things. Cognition is here, memory is here. But this is much closer to what we understand today. Someone who had a huge impact on the idea of perception, and particularly on understanding or investigating illusions, is the philosopher from the 17th century and 18th century, George Berkeley. Berkeley wrote an incredibly influential book that's still a good read today, called An Essay Towards a New Theory of Vision. And so it's a very descriptive book So he talks about what you feel when you see things, and tries to understand in a sort of a pseudo-scientific manner what may be going on. And for George Berkeley, everything was internal. There was no external world, so he's called an idealist. And so the only thing that you have is perception. And importantly, from Berkeley, I think we get one of the first ideas of, Thomas Bayes wasn't alive at this point, but what is sort of the Bayesian idea that actually what vision is it's rather than the sense itself, things coming from the outside world. It's an act of judgment grounded on your experience. So all of this is sort of philosophy, and here, when we get into the 19th century, we start to get into what I think is now clearly experimental psychology. So for the study of illusions, Jan Purkinje, who was a Czech scientist, is incredibly influential, and he wrote his PhD, which was published in 1819, and in that PhD he talks very specifically about how to study subjective phenomenal experiences. So he calls it the subjective sensory phenomena. And he goes through a whole group of these phenomena. He went on to do multiple things. So he contributed to many fields. So, for example, the Purkinje cells in the cerebellum are named after him, and there are Purkinje fibers in the heart. He went on to make very useful contributions to anatomy, physiology, pharmacology. But his real first start was in the trying to objectively study subjective states like illusions. And so, he optimistically said it's an imperative belief of the natural scientist that each and every modification of a subjective state in the sphere of the senses corresponds to an objective state. So what he's doing is, philosophers have been arguing for thousands of years about external versus internal truth. And so what Purkinje does is reformulates this problem into a much more straightforward problem. So it's not internal versus external, but actually it's psychology versus physiology. And this really was the vanguard of a whole group of scientists who started to study subjective experiences in a sort of an objective way. In particular, I want to highlight Gustav Fechner, who's sort of the grandfather or the great-great-grandfather of psychophysics. He was the first person to really come up with a sort of a scientific method, which is, still largely what we do today, where he was working with things like just notable differences and measuring psychophysical thresholds. And the sort of the zeitgeist, the sort of the sensation at this time can be very well summarized by Hermann von Helmholtz, who, as always, is very elegant in his language. So what we see is the solution to a computational problem. Our brains compute the most likely causes from the photon absorptions within our eyes. So this really grounds subjective experience deep within physiology and sort of cognitive psychology. So for Helmholtz, illusions are sort of neurocomputational solutions that don't correlate with the photon absorptions. It's a valid solution. It's something that the brain does, and it can be studied just as everything else can be studied. So in psychophysics, cognitive psychology, we have these affordances between physical characteristics in the external world and internal psychological properties. So, for example, wavelength as a physical property has a perceptual affordance, which is color. Luminance has a perceptual affordance, which is brightness. And contour contrast has a perceptual affordance, which is shape. And similar affordances can actually apply just as well to illusory experiences. So again, for wavelength, you can have things like color filling in, or there are a large number of color illusions. For luminance, for example, after images, something that I'm going to talk about today, because I have some data that's relevant to that, and contour contrast, which the illusory example is something like the illusory contour, which I may also briefly show. So physiologists started to get interested in this and wanted to start recording neurons and working at what neurons are doing. So, for physical stimuli, that's fairly straightforward. But people started working pretty early on looking at non-veridical, so, sort of non-physical or illusory type stimuli. And perhaps the most famous and really beautiful study was published in 1993 by Peter Hans and von der Heydt. And so what they were doing is, in the awake monkey, they were mapping cells in V2 of the visual cortex. And they used, for example, a physical stimulus. So here is a physical bar passing through a receptive field. They then just provided the ends of this contour. And again, the neuron responded, to the same orientation. And then they used what's called an IC, illusory contour. So this is a contour now. So the V2 cell has an orientation shooting preference. So it likes this orientation. It won't respond to something that's orthogonal to it. And so you have this orthogonal grating that's got this series of little gaps. And the little gaps infer that there's a contour there, but there is actually no physical contour there. It's just a sort of an illusory contour that's generated by the line terminations. And they found that the neuron in V2 still responds. It still responds with its preferred orientation to the illusory contour, ignoring the orthogonal non-preferred contour lines. And more recently, there's been a sort of a big push to try to see whether animals respond in terms of psychophysics to illusions. So, not only recording from the brain while you present an illusion, but actually see, what the psychophysical response is. And actually people have found that almost all species, so including the fruit fly, fish, reptiles, cats, including lions and tigers, and non-human primates and human primates, us, respond to visual illusions in a manner that's consistent with the way that they're perceived and consistent with the way humans perceive. So here is a very famous illusion. It's called a rotating snakes illusion, created by a Japanese scientist called Akoshi Kitaoka. And if you move your eyes around, what you should see is sort of shimmering rotation. You should see these sort of circles rotating leftwards or rightwards, depending on which circle it is. And cats perceive just exactly the same thing. So here's a sort of a video of a cat, trying to strike the center of these illusory circles. So he can see that there's something going on, there's some motion. And this illusion has been shown to also affect non-human primates. And so the idea now is that we can interrogate the neural mechanisms that underlie some of these illusions. So we have a series of core questions. And so the fundamental question is how does the neural circuitry in terms of either, the anatomy, where the neuron is, or its physiology, what its response structures are like, its receptive field properties, how they represent and impact the resultant subjective perception of illusions. What are the correlations between the neuronal response properties and the perceptual results for illusory stimuli. So effectively, the physical stimulus doesn't get you to an illusory perception, so you need to have something else that's going on. And that's why I call these things neuronal priors. So there's something about the visual system that it's learned a series of tricks or rules, And those tricks or rules normally work very well, but in the case of illusions, effectively, there's a sort of a chink in the armour, that there's a sort of a disconnect between what the system would normally do and what it does under the illusion. And so we work with humans, with cats, and with monkeys. So all three of these species are highly visual. The cat is a very well-known model for visual processing, as is the monkey. And the illusions that I'm going to talk about today, and there are many illusions, and it's maybe something we can talk about a little bit in the panel. But we'll focus on what I would consider some of the more bottom-up type of illusions. So these are illusions that we think probably have a very clear mechanism that can be explained somehow in defined areas of the brain. So the first illusion is the negative afterimage, and so this is an illusion which is basically to do with luminance and form. The second illusion is called the pin illusion, and this is an illusion of complex flow motion. So this is three-dimensional navigation through the world. I'll briefly go through some of the DNA modelling we've done on the pin illusion, and if we get time, I'll cover the serpentine illusion, although that one we've only published stuff in humans, so we don't have any neural data, just models. So before I start, I do want to say that most of this work is founded in a long-term collaboration with my very good friend, Dr. Wei Wang, who is also an investigator at this institute. So I think a little bit like Maurice and Ali, Wei was a postdoc in the lab that I did my PhD, and I spent many years with him. We used to do acute electrophysiology experiments. So in an acute experiment, normally you have to stay in the lab somewhere between 4 to 8 days. So you just live with someone for effectively for a week, 24 hours a day. And so that was the sort of the foundation of a very long and fruitful collaboration with Wei. And Wei also contacted a wonderful senior, so he's 83 or 84, but still very active vision scientist called Lothar Spillman. So he's very famous for being the editor of a very influential book on visual neurophysiology in the 1990s, and most of his work is also looking at the interface between perception and neural mechanisms, and so he's been involved in almost all of the work. And so although he's 83 or 84, he has the energy of a 20-year-old, and the passion is unabated with age. Yeah, there's Lothar. Okay, so the first set of illusions are after images. So the first quantitative description of the afterimage was published in Nature in 1897 by a guy called Bidwell, and effectively he had a light which was connected to a rotating disc, and so it would sort of rotate, and people would see when they looked at it, not immediately, they needed a little bit of time to see it, but there was a ghost that would follow the light around. And the colour of the ghost was opposite to the light itself, and so this is the first description in the literature of a negative afterimage. And so here's an example. So please fixate here, and if you maintain fixation, what you should see after a few seconds is a green spot that's going round clockwise, and in fact there is no green spot there. So effectively all that's happening is that I'm turning the reddish dots on and off on each frame. So I've created an animation, but there is no dot that's moving around. We're only taking one dot away on each frame. And so this is with colour. this is sort of a fast-moving, dynamic stimulus. But actually what we're interested in trying to study is the classical negative afterimage. And so what our stimulus is it's just a bright or a dark disk that's presented for a number of seconds, depending on whether it's in the human or the cat. And the question that we want to ask fundamentally is how these perceptual afterimages that are thought to be dependent on low-level neural responses correlate or affected by the neural properties of the neural cells themselves in low-level visual areas. So here's just an example of the sort of early visual pathway. And so retina, LGN, and cortex. And the question we have here is that there are some fundamental properties of this pathway. So one of the most fundamental is one which effectively is started at the very first synapse in the retina, and that is the bifurcation or the separation into the on and the off pathway. So this is a really beautiful anatomical distinction, and so you have a very, very beautiful circuitry in the retina, which basically bifurcates into on and off bipolar cells and on and off ganglion cells. They then basically project forwards into the LGN, and they maintain this separation between on and off. And so these channels are critically important for vision. But interestingly, no one has really studied whether the on and off system differentially affect afterimages. So for physical stimuli, so here, we actually know that there's an asymmetry in perception between bright and dark. So in general, humans are better at seeing dark than bright stimuli and see them faster or more quickly. And so in the retina, there's a sort of more off cells, and this off dominance is propagated through thalamus into visual cortex. And this makes the visual system more sensitive to shadows or, decrements, decreases in luminance compared to increments in luminance. And so this is a very well-known phenomenon, but no one has really studied to see whether this very, very fundamental channel distinction causes differences between bright and dark afterimages. And if so, how they relate to the neuronal firing properties. So, okay, let's go through this. This is important to understand the data. So here we have an on cell. An on cell likes light, an increase in light in the center of its receptive field. So if a stimulus is light and is turned on in its receptive field, the neuron fires with an increased number of spikes. And then the firing rate goes down when the stimulus is removed. If the same neuron is presented with an opposite sign stimulus, so in this case this is an on cell, and it's presented with a dark disk, so it's non-preferred stimulus, the neuron will be suppressed, it will reduce its firing rate. But when you take the disk away, there's a rebound or a burst response. And so what you see here is that there's a neuronal firing pattern here when there is no stimulus present. And that's very similar to when you have a physical stimulus and you remove the physical stimulus and you have the perceptual afterimage. And so what we wanted to do was to see, and people have linked the offset responses with perceptual afterimages. In addition, adaptation is also a sort of an important factor from the retina onwards, and adaptation will play into the mechanisms that generate these responses. So likewise, for an off cell, it has a preference. Okay, so likewise for the off cell, you can see here that the preferred stimulus creates an increase in firing, and opposite sign stimulus causes a suppression, and on removal of the opposite sign stimulus, there's a firing rate increase. So the question is that given the known difference between off and on responses for physical stimuli, whether those differences are observed in perceptual afterimages. So in this case, a bright image creates a dark perceptual afterimage, and a dark image creates a bright afterimage. And then the question is that, in this case you have, this is a bright image, so it suppresses the off cell, and when the off cell, when this is removed, then there's a firing rate increase here, and so this sign is the same as the perceptual afterimage here. And the question is whether there are differences between bright and dark afterimages and on and off cells. So first we did psychophysics in human. So both this study and the next study use a pedestal. So this is really a very, very good way to try to quantify psychophysically these sorts of subjective phenomena. And so the paradigm is as follows. You present a physical stimulus for about 4 seconds. So this basically causes the afterimage. So if you just show this disc for 4 seconds and then remove the disc, keep the subject fixated, they'll report a dark afterimage. Okay? And what we do is, after this 4 seconds that the stimulus is on, the stimulus is turned off, we then inject a pedestal that's going to be basically the same brightness as the afterimage. And what we want to do is to try to play with the pedestal, change its contrast until we null or negate the afterimage. So we cancel out the afterimage perception. So the subject basically just says that there's no afterimage and there's no pedestal. They just see the grey background of the screen. And because afterimages basically swell and die over time, we then put a mask after 350 milliseconds so that we really target this initial phase of the response, which is going to be the time where the neural responses are most clearly comparable. And so the question is, if you have your afterimage and your pedestal, what contrast of pedestal do you null or negate the afterimage? And so these are our individual subject responses, and this is the population psychophysical curve. And so what you can see here is that the black stimulus, which is a bright afterimage, requires has a higher pedestal contrast to create a null. So the 0 here means that the subject didn't see either the afterimage or the pedestal, and so the afterimage was effectively erased from conscious perception. And so this requires a pedestal contrast of basically around 0.14, compared to the white stimulus or the dark afterimage, which has a pedestal contrast requirement of around 0.9. So there's this difference. And this is just the population psychometric curve. We also fitted the curves using a Bayesian analysis, and so we could calculate the posterior distributions plus or minus the 95% confidence intervals, just to show that there are changes in both the threshold and the psychometric slope. We also tested whether the duration here has an impact. And so in this case, we used basically bright and dark stimuli. And so what you can see here is the responses. So the paradigm is you show physical stimulus, bright or dark. It then turns off. This is now the afterimage, so this isn't really what's on the screen, it's just what the subject perceives. And they tell us how long the afterimage is visible until it basically disappears. And again, there's this difference between the dark and the bright stimuli, or the bright and dark after images. And so there's it's sort of a very significant difference, the higher stimulus contrasts when comparing between the dark and the bright stimuli for longer stimulus durations. We also found that there's a difference in the latency, and so the latency again is different for the bright versus dark after images. I'm not going to talk about that because it's in the paper. The details are available if you want to discuss them later. So now we move on to the neural responses. So this is recorded in cat. This is an anaesthetized cat. And so there's no perception going on here. And so that's a potential flaw. But what we're really interested in at the moment is trying to understand whether the sign and the characteristics of the neural responses are consistent with the asymmetries that we saw in the human psychophysics. So again, here we have an example of an off cell responding to its preferred stimulus. So it responds when the stimulus is on, and when the stimulus is taken away, it basically reduces its firing. And for an on cell, the on cell is basically suppressed, and when the off stimulus, the dark stimulus, is taken away, then there's this rebound firing that's observed. And likewise for the opposite polarity, so the LGN on cell prefers this white stimulus, the off cell is suppressed by it, and when the stimulus is removed, there's a rebound response. And so what we found was that there was indeed an asymmetry. So the on cells had a significantly higher firing rate, both in terms of the peak response and then basically this sort of sustained response. So this is a stimulus duration of 500 milliseconds. So it's somewhat shorter than the stimulus presentation for the human psychophysics, and so you'd expect a slightly weaker effect. But there is nevertheless a significant difference in the firing rate between the on cells and the off cells. And that is consistent with the psychophysics in that basically it was the dark stimuli that caused, that needed the stronger contrast pedestal. And so the sign is the same for both the psychophysics and for the physiology. And this is just basically the population data showing that effectively we're seeing the same thing between the two. And we also have the ability to measure S potentials. So you can see here there are these little pre-potentials. So here's our spike and here are the pre-potentials. And these pre-potentials have been shown to come from the retina. So they are effectively retinal ganglion cell inputs. And so we can measure whether the retina is also affected or not by this difference. And indeed, the pattern is the same. So effectively, the on cells have a larger firing rate response, which was also significant. And we showed that there was an asymmetry for duration. And if we keep recording for longer, so we present the stimulus for a longer time. So in this case now, we're comparing a stimulus that was presented for 300 milliseconds versus one that was presented for 4,000 milliseconds. And we then look at the response. This is an on cell. You can see that there's elevation of firing for the peak, but more clearly for this long duration tail. So even after 1,000 milliseconds after the stimulus was removed, there's this elevated firing that's maintained. And this was the case for the on cells, but not for the off cells. And this is just another way to show the same data. So the interim summary here is that the bright after images, whose putative origin is the on rebound response, are stronger than dark after images. And the on cells show stronger post-stimulus rebound responses, so that's consistent. bright after images are perceived for longer, and likewise on cells show longer duration elevations of firing, and the latency differences are also the same. So the next question is whether, if we now look at another fundamental feature of the receptive field, this is the centers around antagonism, whether that is also going to play out between the psychophysics and the neurophysiological recording. So neurons in the visual system are size tuned. If you put a small stimulus in the receptive field, there's not much response. You make it slightly larger, there's effectively a peak, an optimal response, and you make the stimulus any larger, then you get a suppression. And so, this is very well known and established. But the question is whether for our opposite polarity stimuli and for the psychophysics, whether there's a size tuning of the perceptual psychophysics and the neuronal post-stimulus responses. The previous work was published in 2017, and this has just been submitted. Here's our classic response to a stimulus, and there are differences between on and off cells, but I don't really want to focus on the stimulus responses, but on the post-stimulus responses. So here now are the post-stimulus responses. So you can see that there's a suppression for both on and off cells when the stimulus is presented. This is the non-preferred stimulus, so for an on cell it's the dark stimulus, for an off cell it's the bright stimulus, and then there's this rebound response firing. And you can see that actually, although there's no stimulus on the screen, the rebound response is also size tuned, so it's basically got this preference. So here's the size tuning. And again, what you can see is that the response rate is significantly elevated for the on cells compared to the off cells, and also for the S potentials, so the retinal inputs. And there's a much clearer difference in the response suppression. So this curve is basically tracking the response here, and so there's more suppression for the on cells before this rebound response. And so we know very well that basically a greater amount of inhibition will cause a greater amount of rebound response firing. So all of that looks very, very consistent. And so there are these differences now in the size tuning for the on and the off cells. So this is the size tuning of a non-existing stimulus, the post-stimulus response. And so the on cells have larger receptive fields and they have weaker suppression. So this is opposite to the stimulus responses. So there's something about removing the stimulus that sort of flips the on and the off cells around. Okay, so to wrap this up, this section at least, here we have a firing rate-based network model of the LGN that was published recently by Moberhan et al. And so they basically have a sort of a mechanistic model that includes retinal ganglion cells, LGN cells, and cortex. We ignore the cortex. We're only interested in this section. And if we look just at the post-stimulus responses, we fit the model to the population data. So this is the data and this is the model. But then we actually have a look at the size tuning just based on the parameters from here. And we find that what I'll do is I'll just give a very brief summary of this illusion, the Pinner illusion, and the neural mechanisms, and then we can maybe discuss in more detail in the panel. So in this illusion, expanding and contracting pattern creates an illusion of rotation. And so you can see here that actually these disks seem to be rotating clockwise or counterclockwise. And if we take exactly the same pattern and we counter or clockwise rotate it, we see expansion and contraction. And so this illusion is a very interesting illusion which involves complex flow motion. So this is motion that's normally occurring when you're navigating through the visual world. So you get basically this expansion, contraction, clockwise or counterclockwise rotation, and a combination of these creates a sort of a spiral motion. And so the beautiful thing about the Pinner illusion is that physical motion produces consistent illusory motion. And so physical contraction produces clockwise rotation. Physical expansion causes counterclockwise rotation, and likewise for the physical rotation, creating the illusory radial motion. So we did psychophysics in human, showing that the psychophysics of this illusion is just very, very clear. It's almost 100% for all subjects. So it's very, very clear under which circumstances we see which forms of illusory motion. So we did now psychophysics in humans and monkeys. So these are awake monkeys, and they're doing exactly the same psychophysical task as the human. And so the psychophysical task is basically that you have this stimulus that's either expanding, contracting, or rotating, and you have to choose whether it's clockwise or counterclockwise for expansion or contraction. So there's a 3 alternate force choice detection task and a 2 alternate force choice discrimination task. And to cut to the chase, effectively the psychometric curves, and we're using the same sort of nulling paradigm, so we're adding in physical motion to try to null the illusory motion, and we see that these curves are shifted for the illusory conditions, consistent with the model that we've generated from the psychophysics, both in the human and in the monkey. And this is the first demonstration, I think, in the literature of actually measuring the magnitude of the illusory percept in the monkey. So people have shown that monkeys respond to illusions, but no one's actually measured what the magnitude is. And effectively, the magnitude is more or less the same, or actually even in some cases slightly larger in the monkey than it is in the human. We record them from MST neurons, and we find that basically, again, according to our prediction, we see these enhancements of responses for the illusory motion. So for neurons which prefer rotation, they respond when there's only physical expansion and contraction, but there is illusory rotation in a manner that's basically consistent with the fact that they're responding to the illusion. The population responses are very clear. They show very clear distributions. And I'm going to cut through a lot of this to basically just get the take-home here. The take-home for this illusion is also very clear, and that's that the MST neurons respond to the illusion almost exactly as they would respond to physical complex flow motion. So the MST neurons respond with exactly the same tuning, although they're delayed by around 15 milliseconds. So their response to the illusion is about 15 milliseconds later than their response to physical motion. And we've done some modeling of that using deep convolutional neural network, and we can also discuss that in the panel. Okay, so with that, I would like to thank you, and I'm happy to take questions if there's time. And a special thanks to my collaborators, people that worked on each of those projects, and also the funders. Thank you very much.
So maybe I take this opportunity to ask a general question. So it was one of the things that was striking to me was how big was the firing of the neurons to the after image, the on-center and off-center neurons. I'm not sure, was it recorded in LGN? I think the ones that you were showing.
That's in LGN, yes.
So I think it was a slide 28 or something, and the size of the firing after the offset was even bigger than the response of the other group of neurons to the onset of the stimulus.
Yes.
So which is very interesting, because I know we all knew about this offset response, but I never knew that the magnitudes can match the onset response for the other group of cells. But my question is something else. I wanted to know your take or your thoughts on why does the visual system have such a strong should have implemented such a strong after image mechanism. I mean, you could have thought of a mechanism a circuit mechanism that suppresses these, off response when you turn off the stimulus responses. What do you think? Is there any evolutionary benefits to the animal to have something like this? Why should we have something like this?
So, I mean, I think you can approach that question in a couple of ways, sort of a bottom-up versus a top-down approach. The bottom-up approach is basically just to do with adaptation, right? So, as far as we know, almost every synapse in the brain adapts to some degree. And so adaptation is a mechanism that, basically is found everywhere. And it's obviously, in a way, very, very important on a number of levels. So, as an example, in terms of encoding efficiency, adaptation is very, very useful because basically there's no point in constantly responding to the same stimulus. And so you basically reduce your firing rate down. If you add into that the idea what the brain is very, very interested in is change or differences, you combine adaptation with the ability to care or discriminate temporal changes, then I think the after image, these post-stimulus rebound responses are really part of that process. So that's sort of the bottom-up description. The top-down description is really, again, to say that what you can imagine is going on, again, just with a sort of a Bayesian prior, that effectively what happens is that you want to reduce the response of neurons, and you're only interested in the surprise or the difference or the error from your prediction. And so, sort of an orthogonal way to look at the same thing, and those two things do overlap, but an orthogonal way to look at it is to take a predictive coding approach and just imagine that actually, again, that those rebound responses are a sort of a byproduct of, the sorts of things that you need to do to reliably predict changes or differences in the world and then update your model, as it were. That's a sort of a two orthogonal pronged approach to answering your question.
Okay, thank you very much.